Welcome, everybody. I'm Paul Fletcher from the Department of Psychiatry, and I'm really pleased to introduce Michael Frank. It's fantastic to have someone like Michael Frank speaking to us. He's currently the Edgar L. Marston Professor in the Department of Cognitive, Linguistic and Psychological Science at Brown University. And his original PhD in Colorado was in psychology and neuroscience. And in the interim, he's also held a professorship in Arizona. For me, Michael is really a translational scientist par excellence. His basic interests are in both computational and experimental approaches to reinforcement learning and decision making. And he's really applied these in incredibly innovative ways and neurobiologically informed ways to neuropsychiatric illnesses, and has offered important findings that are truly translational. And he's rightly won a number of prestigious awards for his work. Today, his topic is striatal dopamine computations in learning about agency. So, Michael, over to you.
Well, thank you so much, Paul and Valerie, and thanks for inviting me. I look forward to getting some feedback and talking to you guys. Okay, so I'm going to start off right away with the idea that has held some prominence for a few decades about how dopamine might be involved in some form of reward, or rather reward prediction error. And so I'm sure many of you are familiar with this, given many of the early findings from Wolf and Schultz's labs and others, where if you have, in this case, a monkey, and you pair a stimulus like a light or a tone that then is followed by a reward a couple of seconds later, if that reward is not expected, you see an increase in the firing of midbrain dopamine neurons. And initially, people might think, okay, that's consistent with dopamine encoding some kind of reward value. But then if the monkey is trained for a while in a Pavlovian association, such that they know that this stimulus actually predicts the reward, after that training, then you see that dopamine no longer cares about the reward itself when it's delivered, even though the monkey is perfectly happy to get the reward and enjoys it. But you see there that dopamine does show an increase in activity to the stimulus, which previously was perfectly neutral, but which predicts future reward. And then if you play a trick on the monkey and you give the stimulus, but you then withhold the reward, then you tend to see a drop in dopamine levels. And so that kind of finding, which has been supported in many other experiments that have manipulated all sorts of things to test the general idea, and also across species, has been taken to be in support of evidence that rather than coding for something like reward or directly movement or something, this dopamine signal is coding for an error in the prediction of reward. So here the reward is unexpected, and here it's expected, so there's no prediction error, but the stimulus, which was itself previously unexpected, is now suddenly a prediction of future reward. So there's an error in how much the animal expects reward as soon as the stimulus arrives. And here, that's even more evident because the animal didn't expect reward, now it does, and now it's expecting reward for a while, and it's expecting it to occur at a particular time, and then when it doesn't occur, there's a sort of disappointment, and that's reflected in a negative reward prediction error signal. And one of the things that I and other people have been interested in is, given that there are these reward prediction error signals that come from the midbrain, how could that impact activity and learning and behavior in the downstream circuitry that responds to dopamine in the striatum? So the striatum is the area of the brain that receives the most amounts of dopamine, and its dopamine function in that region is very well studied. And I'm not going to go into any detail on this here, but the general idea is that there are these different pathways that go through the basal ganglia that have different receptors for dopamine, and that the impact of dopamine increases, like reward prediction errors, is different in these two pathways. So one pathway is involved in sort of go activity or facilitating actions that are related to motivational value, and another pathway is involved in suppressing those actions that are unwanted or produce negative rewards. And so the general idea here was that these reward prediction errors can be married with this sort of circuit level model of the frontal striatal circuit that says when you get reward prediction errors, you sort of potentiate neurons in the go pathway and you depotentiate neurons in the no-go pathway, whereas when you get negative prediction errors, it's the opposite. And that essentially is an opponent model that says that, you know, dopamine can go up and down, but it induces learning in opposite directions in these two different pathways. And we think that there's some reason the brain might have come to that solution, but we're also particularly interested in how that might relate to findings in behavior, in this case in clinical populations. So some of my very early PhD work on this was looking at patients with Parkinson's disease who have low levels of dopamine. They have depletions of the neurons that make dopamine in the midbrain. And we gave them a task that requires them to undergo reinforcement learning. So they had to learn to choose certain stimuli on computers that lead to high probability of positive, you know, winning points or winning money, whatever. And then other stimuli had a high probability of losing points. And what we found there is that Parkinson's patients who have low dopamine and are off their medication are actually just fine at learning to avoid those actions that led to lots of negative reward prediction errors. And that, according to this sort of theoretical framework here, depends on having low levels of dopamine to learn from. Whereas they're impaired at learning to choose those actions that lead to high levels of positive reward prediction errors. And then you can ask, is that really related to dopamine by just increasing their dopamine levels with their regular dopamine medications? And then you see that those patients that used to be worse at learning from positive prediction errors are now better there, but they actually are worse at learning from negative prediction errors. And that was consistent with a model that we built of this circuit, because normally your brain wants these dopamine levels to go down when you get negative prediction errors, but the medications continually stimulate these D2 receptors that prevent those neurons from learning from negative prediction errors. So that was the general idea. And just to say that that was our original finding, but that basic finding has been replicated now in more than 15 different studies in our lab and many other labs with these kinds of tasks and other tasks, including some that Valerie Voon did a while ago, about 10 years ago. This is another study recently from another group showing how the effect of medication in Parkinson's disease is effectively to change a parameter from a reinforcement learning model that symbolizes how much people learn from losses or negative prediction errors as opposed to positive prediction errors, and showing that the medication effect is to essentially reduce learning from negative prediction errors, similar to this effect here. And they also showed in imaging that that was manifest by changes in the striatal response to negative prediction errors in ways that are consistent with this general model. And we think that even though it's a somewhat contrived lab task to assess this function, we think that it has some clinical relevance, because we know that Parkinson's patients, at least a subset of them, when they're on medication, can develop some impulsivity disorders like pathological gambling and other things, something that Valerie has studied a lot. And you can imagine that while this might not be the only mechanism that is related to this, if you have a bias in your brain to learn more from positive than negative prediction errors, then, for example, playing blackjack, which has a 48% chance of winning, now your brain interprets it as if it has a 60% chance of winning or something like that. And on the flip side of that, the off medication state, where there's this other bias in the opposite direction, should be associated with a bias to learn to avoid those actions that lead to negative outcomes. But if your brain is constantly telling you that there are negative prediction errors, then you might be biased to learn to avoid selecting actions even when they actually are good actions. We have some work in rodent models that suggests something like that is actually happening, that supports the progression of Parkinson-like symptoms in animals. Even if you don't further exhibit any more dopamine depletion, this sort of aberrant learning process can itself lead to progression of symptoms. And similarly, we think that antipsychotics and schizophrenia can lead to something like that in the sort of motor symptoms of schizophrenia. And this is just to show that this cartoon diagram of the basal ganglia is supplanted by another cartoon diagram that we use to simulate this circuit in more detail with neural network models that I'm not going to go into any detail, but I just wanted to mention that we try to use these sort of relatively detailed circuit level models to ask questions about how can you account for the basal ganglia dopamine circuitry, and how differences in activity in these different populations might relate to differences in learning and behavior. And that kind of model predicts this kind of pattern of data and other things. But I want to move beyond that. And before I go to the sort of the main topic of the talk, I wanted to also emphasize that everything I just emphasized was on the learning side, that you have these prediction errors that go up and down, but it's also well studied for a long time that dopamine also directly affects performance or motivation. And we think it does so via very similar mechanisms by modulating essentially the degree to which these striatal D1 and D2 receptors represent the costs and the benefits of actions. And so in this paper, in a collaboration with Roshan Cools and postdoc Andrew Westbrook last year, he studied how people engage in decisions as to whether to perform difficult cognitive tasks or not. And Andrew has this procedure where he asks, would you rather do a difficult working memory task? In this case, it's the NBAC task that is hard, or an easy one. And generally people like to do the easier one over the hard one. It's really not fun to do a task that's really challenging. And so you can get them to prefer the hard task if you give them enough money to do so. So you can pay them, in this case, €4 versus 0, or, and then he'll give people, different choice options for different amounts of money. And then you can construct sort of an indifference curve of how much money somebody is willing to, it needs to get in order to do a harder task. And so what I'm showing on the right here is predictions from this model. It's a sort of a more simplified version of that complex neural network model that I mentioned, where if you plot on this graph here, the dotted vertical line here is the indifference point. So that would be like, if you're not sure whether you should choose the easy task or the hard task, there's the benefit, which is how much more money would you get for doing the hard task. And then there's the cost, which is how hard it is, how subjectively effortful it feels. And so as you go to, sorry, we're missing the label here, but you'll see that in a minute. The point is that as you go to the right of this indifference point, the model predicts that, the benefits outweigh the costs. And so you should increasingly choose the hard task. And as you go to the left, you should increasingly choose the easy task. But the more interesting prediction is that as you simulate increases in dopamine in this model, which is reflected by this ratio here, it predicts that the steepness of this curve goes up and up, so that with more dopamine, you're more likely to weight the value, the reward benefit of performing the hard task. And you should say, okay, I'm going to just choose that hard task more consistently. Whereas if you have lower levels of dopamine, you still go up, but not at strong a rate. And then because this model has this opponency where there's like the benefits and the costs on opposite sides of these indifference points, it predicts an asymmetry in that choice function, so that the more likely you are, if you have high levels of dopamine, you actually don't represent that cost as strongly. And so this is a shallower choice function, whereas low levels of dopamine allow you to actually discount that costs a lot. So this sort of asymmetry in the slope is what is a core prediction of this dopamine model. If you apply it to making decisions, and these decisions in this case happen to be about cognitive actions, but the same thing would apply for motor actions and physical effort and so forth. And what Andrew saw was that there's evidence from, in this case, PET. If you look at dopamine levels in the striatum and the caudate, people with the highest levels of dopamine exhibited the steepest choice functions to the right of the indifference curve and the shallowest functions to the left of the indifference curve. And then if you gave people a dose of methylphenidate or Ritalin, which has several effects, but one of them is to increase striatal dopamine, then you could see that its effect is equivalent to what you would predict from increasing dopamine levels in this model, where people are more willing to exert more effort for the harder task as the benefit goes up. And they also show shallower functions relative to placebo as you go to the left. And so my point here is just to say that that you can use this model to account for asymmetries in cost-benefit decision-making, and in this case, apply it to cognitive effort. And we also think that that's potentially interesting because, as you all know, a lot of people take these smart drugs like methylphenidate and Adderall and others to improve their cognitive performance. And this study suggests that while it might improve performance in some cases, it might be actually increasing people's willingness to engage in cognitive effort rather than their ability. Okay, so that was a very brief tour of a big literature really actually on reward prediction errors on learning and performance. But the biggest part of this talk I want to talk about going beyond sort of the notion that dopamine just does one thing all over the striatum or all over the brain. And so we know actually now, over the last couple of decades that there are diverse dopamine dynamics in different target regions. And sometimes dopamine signals, when you record from them in the concentration levels, don't show just transients like reward prediction errors, but they show things like ramping. We know from Josh Burke's lab that dopamine in target regions like the striatum can sometimes even be decoupled from spiking at the midbrain cell body, which suggests there might be some local regulation of dopamine at the level of the striatum. And this third point is one that I essentially just motivated on the last slide, that there's a role for dopamine directly in movement and motivation separate from learning. But from the computational level, this notion that there are reward prediction errors has been incredibly useful for understanding all sorts of things about dopamine and behavior. But if you take it at face value that that's all it's doing, it's an impoverished signal if you want to learn in the brain how to assign credit to which circuits in your brain actually generated rewards. So the idea is when you're making a decision, let's say to get a cup of coffee and drink it and it's delicious, you get a reward prediction error, but should you reinforce the decision to get the coffee, or should you reinforce, for example, the low-level motor program and the awkward reaching movement that you might have taken to get it? And the problem is, while most models, including our own models of dopamine in the basal ganglia, suggest that the way you sort of credit the striatal neurons that cause the reward is to simply combine dopamine levels with activity. So neurons that are active are the ones that learn. But in the real situations, lots of neurons are active at the same time, and they may not all be contributing to behavior. And you have many different corticostriatal circuits for, let's say, reaching or higher level decisions. So how do you know, how does the brain know to reinforce the right ones that are like the most efficient ones to then be more likely to get reward in the future? And I'm particularly, because we're going to be studying this in a rodent model, we're going to use a simplified design that will ask, is the thing that you should credit the agent's actions themselves? So it's not this coffee example, but just whether the agent or the animal, the person, is in control of the rewards or not. And I'll try to make that more clear in a minute. To motivate that, I just want to use an example that I like, because I like snow sports. And so here you have these two kids going down the hill sledding, and they're having lots of fun, and they might be getting all sorts of reward prediction errors. But they're not actually causally responsible for that, other than their decision to get in the sled in the first place. But here is a mogul skier who's going down the hill, and in order to experience positive reward prediction errors, they have to tightly coordinate their action planning with particular states of the environment that then lead to the reward prediction errors, and they are responsible in their choices for good outcomes. And if you don't like that example, another example would be guitar playing. So some people really love to listen to people playing guitar, and you might experience a lot of reward prediction errors. But that doesn't necessarily mean that you know which actions to play yourself if you're to be given a guitar. And so to illustrate the sort of problem at the circuit level here, we have these, this is a cartoon of one of our sort of hierarchical neural network models of corticostriatal circuits. I'm not going to go into much detail except to say that there's a lower level motor circuit here that is just about selecting low level motor plans, and then there are higher level circuits that are interacting between the prefrontal cortex and the basal ganglia that are involved in selecting higher level goals, rules, task sets in this case. And this might be like the decision to get a coffee, and this might be your reaching action. And the problem is that dopamine from the midbrain innervates all of these levels of the corticostriatal circuitry simultaneously. And so if you made a decision that was that led to reward, you might actually reinforce the wrong circuit, and you might lead to the conclusion that just because you experienced reward here, that you'll know what to do here. And if you did that, it would lead to these sort of disastrous consequences if you have never skied or don't know what to do here. And so a solution to that, if you are building it into a computational model that has to solve problems, not mogul skiing problems, but related complex learning problems, is you can do something like this. You can say, well, I have some mechanism in the brain that detects whether this part of the striatum changed the cortical state or changed the world in some way that therefore might be related to, might have caused the reward. And that if that happens, if you have a way of detecting that, you can use that to amplify the dopamine signal that then goes back to that circuit and less so to the other one. So that's the general idea here, is that if you have some mechanism of detecting which of your circuits was affecting behavior, you can use that to amplify the dopamine signal there. And if you do that in computational models, it helps it learn in a much more efficient way. And we had some, there are some biophysical reasons to suspect that that might work. But I'm going to try to, that mostly it was coming from computational considerations. And so we want to study that. And so the overview of this here is that we think there should be these sort of what we call actor-weighted dopamine signals. So dopamine signals that are weighted by which of your underlying circuits or actors were responsible. And I'll tell you about how we think that in the rodent model actually is related to not exactly what I just said on the last slide. It's not just that you're increasing dopamine to one circuit or another, but it's mediated by differences in the spatial temporal dynamics of that dopamine system, which provides information about agency, whether an animal is responsible for the outcomes or not. And I'll try to tell you that this is manifest by regionally specific dopamine transients that are essentially like reward prediction errors related to agency. And then the beef, or the main part of the talk, shows that there are these waves of dopamine that propagate across the mediolateral axis of the dorsal striatum in a way that we think is particularly adapted to implement this sort of credit assignment to one circuit versus another. And I'll give you a high-level overview of a model that tries to tie together these different dopamine dynamics. Okay, so all this work that I'm going to talk about from here on in was done by a really talented postdoc, Arif Hamid, who's now going to be an assistant professor in Minnesota. And what he's doing here is looking in mice, and he's going to be imaging the axons of dopamine terminals in the striatum, over the dorsal striatum. And he's putting in a cannula so that he could image about 80% of the dorsal striatum at one time. So instead of imaging just a single dopamine neuron or a single level of dopamine in the striatum, he's going to be able to image the activity of many axons at the same time. And he's going to be doing that with GCamp, which allows you to look at the terminal activity of those axons, but also D-Light, which is a way of measuring the concentration of dopamine release directly. And so you'll be looking at plots that look like this with a top-down field of view, where medial striatum is to the left, lateral is to the right, and anterior, posterior that way. Okay, so of course we didn't have mice doing mogul skiing, so I'll have to go over the task of what Arif actually did. He had the animals on a wheel while they're being imaged, and then they experienced these tones, these auditory tone transitions that escalate in frequency as the animal approaches a reward, which is going to be water in this case. And in some of the experiments, he also had these visual stimuli that sort of went along with the tones so that they get some information about their progress to reward. But the trick is that there's sort of two different task designs here, which have sort of hierarchical structure. So in one task, it's instrumental, so that in order for the animal to make these tones move in their frequency and then to get the reward, the animal has to run a certain amount for each tone transition. So sometimes they have to run just a little bit, and sometimes it's far. The distance that they have to run to get the reward is sampled from a uniform distribution. So the animal never really knows at the beginning of the trial how much it has to run. But as it experiences these tone transitions, it could sort of infer, okay, I'm in a task that is allowing the tones to go really rapidly and I'm going to get reward soon, or they have to run a lot. But then in other sessions, or sometimes within the same session, the task has exactly the same structure, but the rewards are unrelated to the animal's behavior. So we call it Pavlovian, where we sample it again from the uniform distribution, but it's not about how much the animal has to run, it's just how long they have to wait between each tone transition. So here, the animal is in control of the outcomes, kind of like the mogul skier, and here they're not, kind of like the sledder. And we have evidence behaviorally that the mice actually do use these tones transitions that they see or hear to give them evidence about how far they are to the reward. And the way that we can see that is if you measure their lick rate, they start licking because they're anticipating the reward, and they lick more and more and more as they get closer to the reward. And they do so in a way that is informed by the tone, or like how far they are in terms of the percentage towards achieving the reward. and not just the raw amount of time or distance that they've run. Okay, so that was all to set up this question of, is there special dopamine signals that go to different parts of the striatum that allow it to reinforce certain things? And so we're going to focus on the division in the striatum in the rodent between the medial and the lateral striatum. And the dorsal medial striatum is traditionally associated with goal-directed behavior, or representing action outcome contingencies, which is one way of thinking about agency. So if your actions are responsible for outcomes, then you're agentic, you're controlling the world. And so we can ask the question is, if the animal is in this instrumental task, do they figure out that they actually are responsible for these tone transitions, despite all the uncertainty about the uniform distributions and so forth? And if so, then perhaps you should reinforce that area of the striatum that is responsible for representing these action outcome contingencies and using that to motivate behavior. Whereas if it's in the Pavlovian task, then perhaps that region should not get that reinforcement, even though the animal expects reward. Okay, so the main finding here is, so I'm going to focus, I'm going to play some videos of what happens in the dopamine signals, first in just the GCAMP or calcium terminals. And what you're going to see is a timeline when it gets to 0 seconds. That's when the animal experienced the reward in the instrumental task. And what you should notice is that there'll be a big sort of white blotch of activity, which would signal increases in dopamine activity when the reward was achieved. But you should see that it doesn't all happen at the same time in all regions within this striatum. So that you saw it once, it's going to play it again. What you can see is that there is a wave of dopamine activity from the medial striatum that propagates towards the lateral striatum in the instrumental task. But what happens in the Pavlovian task, you do the same thing, you can see there's also a wave of activity, but it's going the opposite direction. And so those were just videos of certain sessions, but you can also quantify this in all sorts of different ways. And Arif did to use computer vision methods for quantifying the optical flow of this activity. And this is basically just summarizing the vector fields, showing that on average, when the animal gets reward in the instrumental task, the direction of travel of these waves is from medial to lateral. And in the Pavlovian task, it's the other way. And if you look at what happens in the dopamine signal in the animals before they've learned the task at all, you do see some interesting spatial temporal dynamics during the reward, but they are irregular. They don't go in any clear direction. And so the summary from this slide here is that this spatial temporal trajectory of dopamine activity is learned because it's not there initially. And it's sensitive to task structure in that when the animal's in one task, the wave is going one direction, and when it's in the other task, it goes in the other direction. And we think that that might relate to a way in which the dopamine signal gets information about the task structure that could support credit assignment that I'll try to unpack in the next few slides. So I'm not going to have time to go through all the details of this. This paper was actually just published last week online in Cell with Arif Hamid again as the first author, and in collaboration with Chris Moore's lab at Brown. So one thing we wanted to do, besides just asking whether the waves go in one direction or the other, is say, well, what happens if you have the tasks reverse within the session? So here is the velocity of the animal when they're in the instrumental task. And you can see they're running at relatively high speed. And then at 0 is when we, unbeknownst to the animal, we reverse the task from instrumental to Pavlovian. And you can see they fairly quickly learn after a few trials to stop running as much, although they're still allowed to run. Whereas when they're in the Pavlovian task, it's, you know, they're not running as much. And then when it switches to instrumental, they increase their running. So they're learning the task structure. They're running only when they need to obtain rewards. And so we're going to look at the dopamine waves in these within session reversals in both D-light and GCAMP sessions. So they're basically just different ways of measuring dopamine activity at the terminals or release. They show the same results, but for those who are interested, I'll show them both. And you can see, I'll show you the videos again of what happens at the reward. Here in D-light, you can see they go in opposite directions, again, instrumental and Pavlovian trials, and in GCAMP in green, again, the opposite direction. Slow down for you to see. So this is basically just showing again that we get waves in opposite directions, but also within during this session when they're going back and forth between the different tasks. You can quantify that, it's significant. And here you can look at the dynamics of that. And so what this is showing, actually I'll focus on the left here, is just individual trials when the animal in pink is in the Pavlovian task, where we're just quantifying the flow of the dopamine activity at the time right after the reward. And you can see it's going from lateral to medial, it's going to the left on average. And then here is the reversal. And you can see for the first couple of trials, it actually was still going in that direction. But then after the third trial in this particular session, it starts reversing and going the other way. And you can see that here quantified across animals and sessions. You can quantify the angle of the wave going one direction or the other. And you can see these nice reversals right at the block change that mirror what you see in their behavior. And so I think I can come back to this if there's a question for this, but we have some simulations through reinforcement learning models that could suggest why waves are particularly well suited to support this credit assignment. But I realize I'm going longer than I wanted to, so I'm going to skip this slide and I'll come back to that if you're interested. And I just wanted to take the last couple of minutes to motivate this model that we have that tries to tie together the dopamine dynamics that are happening in the striatum and why and how they can be used to infer agency and to reinforce behavior. So to do that, here we built a model. This is not a detailed neurobiological model, it's a computational level model. So let's say you're an animal in this task, and what you're trying to figure out is the task generated by an instrumental contingency? So is there sort of a distance to run, or do I just need to wait to get the reward? And so let's imagine that in your brain, you have these sort of experts that focus on, oh, I'm in a situation in which I have to run, it's instrumental or not. And then within the instrumental task, you might learn that there are different kinds of contingencies. Like sometimes you need to run 3 centimeters to move each tone, sometimes 5 centimeters, sometimes 8 centimeters. You can think of that as having different sort of motor models, kind of like when you pick up a glass of coffee or water. Sometimes you expect it to be heavy and sometimes light. And within each of these sort of sub-experts, they would have different predictions essentially about if I had to run 5 centimeters per tone, I expect that after the first 5 centimeters, I'm going to hear this tone, and then after the next one, that tone, and so forth. And at the end of the day, what the model has to do is decide, should I run and how fast should I run? And so in this model, we're asking, how can you assign credit to these different sort of experts and sub-experts in a way that will allow it to do the right thing? And the basic idea is that you should accumulate evidence for those sort of sub-experts and then the overall experts that best predict the outcomes or the transitions that you see in the task. Where agency in this case would be reflected by a congruency between your actions and the outcomes. So if, well, maybe I'll make that clear in a second. Let's say you're running here and sometimes you're in a short trial and you have to run a little bit in order to change the tone. Sometimes it's a long trial. You can ask, is there a congruency between how much you've run and when those tone transitions happen? And so at the lowest level of this model, we expect there to be these prediction errors every time a tone transition happens, because even if you predict you're in the instrumental task, you don't really know exactly when the tone transitions are going to happen. So if you're in a short trial, the tone transitions might happen earlier than expected. And if you're a particular sub-expert that expected it to happen, the tone transitions to be longer, you would get a reward prediction error every time the tone transition happened earlier than expected. And the way this works in reinforcement learning is that those go up across time because of something called temporal discounting. Whereas if the trial is longer, you still should see prediction errors unless you're exactly predicting when they're happening, but they should be, smaller and spaced out as the tone transitions happen. And the idea is that you could use these sort of discrepancies between your actions and your outcomes and when the prediction errors happen to make some inferences about which of these sub-experts is most likely explaining the data, is most likely responsible for the world. And so you could use these reward prediction errors for making inferences about agency. And so the way that happens in the model is that these sub-experts will accumulate the evidence for, one of these sub-experts or another. And if you're in a task that requires, let's say running 8 centimeters per tone, then you see the sort of ramping of the activity of the sort of evidence for that sub-expert and decreasing ramps for the other ones. And finally, at the highest level, if you're saying, okay, across all of my little distance experts here, I'm going to, pay attention to the one that's predicting stuff the most. In this case, it might be that one. And how well am I predicting outcomes relative to just weighting, just the time expert? And so what that does, it says in the instrumental task, it would predict a ramping increase because overall across all my sub-experts, I'm predicting the data better than a model that just says I have to wait without running. Whereas if you're in the Pavlovian task, it would actually go the opposite direction in the distance expert because it's not actually responsible for the outcomes. Okay, so after you've done those 3 levels, you would say, okay, I have evidence that I'm in control here. And you can use that to then decide how much to run. And so that if you have evidence that you're in control, then that can give rise to something like a wave that would reinforce the dorsal medial striatum or the distance expert in this model, which then in the model will produce more and more running in the instrumental session and less running in the Pavlovian session. Okay, so I know that was a lot, but here are the key predictions from this model that predicts that there are these different signals that the brain needs to use to compute whether it's responsible for outcomes and whether it's agentic and should reinforce the behavior. At the lowest level, there's these sub-expert reward prediction errors, and now aligning them so that they're reflected by progress towards reward, so percentage of the trial. And then in the middle level, there are these ramps, and the highest level, there are the waves that you've seen already. And that, these should all inform each other. And so what Arif did is he looked in the brain by zooming in the dorsal medial striatum dopamine signals to see if there are evidence for these dynamics at all the levels. And so at the lowest level here, he's using 2-photon imaging to really zoom into individual axon segments. And he found these really intriguing patterns where he saw that there are certain axon segments that show transients to the tone transitions. They look a little bit different than what you see in the model here because they're sort of distributed across different axon segments. But there's one axon segment that shows this prediction error when you go from the first tone to the second tone, or it shows a transient, and that transient is larger when it's in a shorter trial compared to a longer trial as expected from a prediction error. And then there's another axon segment that shows a transient for the second to third transition and so forth, spanning, sort of tiling the whole space. So that was pretty neat, we thought, because you see evidence in different axon segments for these different kinds of prediction errors along the trial. But they still look like reward prediction error like responses, and they have properties of that. And this part I'm going to not really talk too much about, but we also see evidence that at the sort of zoomed out level across the dorsal medial to dorsal lateral striatum, there are these ramping signals that as the animal is approaching the reward, the dopamine signal is ramping up in the instrumental task and it's ramping down in the Pavlovian task. And the steepness of that ramp is greatest as you are most medial in the striatum. So as the animal is most, the region of the striatum that's thought to be most related to goal directedness or agency has the most steep ramping up and down across the different task directions. And then here we can ask, okay, is it actually responsible for changing the wave direction and for reinforcing behavior? So here I'm just showing the velocity of the animal. We know that it runs more in the instrumental task and runs less in the Pavlovian task. And so we can ask, is there evidence for this, these waves of activity being involved in reinforcing agency? And so what Arif did is he asked, if we look at the wave angle on the last trial, if it's going from medial to lateral, which is supposed to reinforce running, is that related to velocity on the next trial? So it's not a performance effect. It's the amount of dopamine that you had on the last trial. It's not really the amount, it's the direction of the wave. And you could see that if you just look the last trial back, the greater the wave is from medial to lateral, the faster the animal runs. And then you see that effect 2 trials ago, and then it sort of goes away in previous trials back. So this sort of pattern of looking at the some neural signal on the last trial and the trial back and the trial back before that is exactly what you want to look for a reinforcement learning effect, where, generally prediction errors have the greatest effect on the last trial and the trial before that and so forth. But here, it's not the standard, just dopamine activity leads to greater learning. It's the wave of dopamine leading to greater running. Okay, so I know that was quite a lot, but a summary of what we're seeing here is there's various different dopamine dynamics in the striatum. I emphasized the waves, but we actually saw 3 different kinds of dynamics. We saw these waves, we saw ramps, and we saw transients. And we think that the transients are like reward prediction errors but in this case, about the task structure. So when the tones happen when you don't expect them, but also you know, a particular region of the striatum is related to agency and other regions are not. And then the ramps can sort of cumulatively accumulate that evidence for agency to decide, you know, whether the animal should work or not, should it run in that trial or not. But really, we think that that allows it to infer whether it's in the instrumental task or the Pavlovian task, which then has a way to guide these waves to reinforce the correct underlying circuit in a way that supports credit assignment. Of course, there's this opens up this finding that Arif had of waves and these dynamics is a novel finding. And so that also opens up many questions about the mechanisms that give rise to these waves and many other experiments that need to be done. But we're excited about this and I look forward to your feedback. Again, if you want to see much more of the details of this at all levels, I refer you to the paper. And with that, I need to thank Arif for pretty much all of that work and my collaborator, Chris Moore, whose lab he sat in to do the rodent work, and my other collaborators for the initial studies I told you about in my lab and funders and so forth. So thank you very much.